Welcome everyone to our talk today on a view of molecules, from molecules to ecosystems, physiological ecology. We're very pleased to have Bill Karasov here with us, a longtime member of the congregation and a distinguished researcher, now emeritus from the University of Wisconsin. He was formerly chairman of the Department of Forest and Wildlife Ecology, and he is an animal physiological ecologist, which means he uses the concepts and tools from physiology to study animal lives in their natural environment and how they adapt to their natural environment. He grew up in Minnesota. No booze in the audience, okay. And he received his undergraduate degree in biology from the University of Minnesota, which has a fine ecology department. Received his PhD at the University of California at Los Angeles and studied deserts there, desert ecology. Not something we see a lot of here. And then did postdoctoral research at the UCLA Medical School. He came to Madison in 1985 and is now a lifer. He has numerous publications, over 200, and wrote a book. He's been a Fulbright Scholar and a Fellow of the American Association for the Advancement of Science. We welcome today Bill Karasov to speak with us and provide a most interesting and educational talk. Thank you all for coming. Happy Mother's Day to all of you. For those who have been here in person, I hope you enjoyed the brunch and you should pick up a rose on your way out. Thank you very much. Bill?
Oh, thank you, Barry, very much for the introduction. Thanks to all of you coming today and also listening in on Zoom. As Barry told you, I'm someone who studies both physiology, which is the way body parts or the body functions, but I'm very much interested also in relationships between animals and their environment and other organisms around them. Ecology is the study of relationships with animals and their habitats. And you might say that the overarching question of people like me who study physiological ecology is, how do wild animals live in their environment? Our work in physiological ecology is very collaborative. And I wish I had a lot more time to share with you more of the kind of work we do and sing praises to the many collaborators I have. I do want to underscore that I don't do this work alone, but I do it typically in collaboration. And I collaborate with people all over the world. Over the past 40 years, I and my 63 graduate students and postdoctoral associates that I trained, we've joined colleagues all over to advance knowledge about wildlife physiology and toxicology. And as examples, I've done a number of studies with colleagues in Israel on mammals, birds, reptiles, even this interesting Egyptian fruit bat. I've worked with colleagues in South Africa on birds, and also colleagues in Australia on this colorful bird called rainbow lorikeets. I've worked in China on monkeys in the Himalayas. And we also do a lot of work here in Wisconsin, which I'm showing up here in the right-hand corner with two of my able assistants, my daughters, because I've been lucky to bring my family with me on many of these trips around the world and working right in Wisconsin. These are Ariella and Talia handling a juvenile bald eagle, which is nearly as big as them. So Barry mentioned I co-authored a book, and the book is about how animals process energy and nutrients and toxins. These are topics we humans are interested in for ourselves as well. I won't have time to talk about energy today. I'm going to talk a bit more about feeding and digestion and how the particular features of animals, their anatomy and their physiology, might determine whether they're sensitive to toxins that they take in their food naturally. And then my second topic, I'll talk about some of our recent work on the impact of man-made toxins, or sometimes called toxicants, on an ecosystem. So to begin, I want to take you far away from here. I want to take you to the Galapagos Islands, which are 600 miles west of the coast of South America. These islands are famous. They're famous partly because Charles Darwin visited these islands on his voyage with the Beagle, and they inspired him to think about and to partly develop the theory of evolution by natural selection. So they're very, very famous for that. For most of us in modern times, they're famous because they're known as a place in the world you can go and really get close to wildlife. Some of the wildlife are seemingly tame there. And you can get incredibly close to creatures that are sometimes not seen anywhere else in the world, and they're quite interesting looking in their behavior. All of the pictures I've shown here are from Corliss's camera. We were just there 2 weeks ago. It was our trip of a lifetime. And we're still jazzed about the things we saw and did. All the pictures except for one is right from Corliss's camera. Which one do you think is not from Corliss's camera, but maybe from my iPhone? I just never understand why people on trips with me or other people don't get into this kind of stuff the way I do. Those are feces of some of these giant tortoises, and they were glorious. On our hikes, we were shown by the Ecuadorian guides a tree which produced a very sweet fruit that looked like apples. However, its common name, they told us, was little apple of death. And according to our guides, and what I learned a bit more when I got back from the trip, is the name comes from modern day Spanish, manzanilla de la muerte, little apple of death. And it refers to the fact that this tree, the manzanilla, is considered one of the most toxic trees in the world. Its fruit is potentially fatal for humans. Curiously, it is a favored food of the two large reptiles on some of the islands in the Galapagos. The land iguana, which are shown here on the left, it's about this big, weighs nearly 20 pounds, big lizard. And then one of the giant tortoises, there's several species there, this is bigger than us, so it could be 400 or 500 pounds. This left us wondering, what is the toxin in this poison apple? And why are some animals seemingly immune to it? Now, naturally occurring toxins are very common in plants, and there are tens of thousands of them. And at this point, you may be worried, OMG, is he going to talk to us about chemistry? Yes, but it's going to be fun and it's going to be easy. And you can complain to me afterwards. It's not. It will enable me to bring you in to see what it's like to be a physiological ecologist. And as I tell my students, we're going to learn just enough, the bare minimum, to be dangerous. So, as you look at this slide, I'm going to show you 3 things here. And I need you to focus on them because you're going to need these tools in just a minute. So, number 1, there are 3 major types of natural plant toxins that are really easy to recognize based on structure. One structure is what's called a 6-carbon ring. I'm going to go to the screen and count out the 6 carbons so you see them. 1, 2, 3, 4, 5, 6. That's a 6-carbon ring, and there's something else hanging off it, but that's a telltale sign of a chemical called a phenol. A second easy structure is this. I'm going to go again to the screen, point to it. It's a carbon with a triple bond to nitrogen, which is shown like that. And the third group I want you to know about also have rings, but they have a nitrogen in the ring. There's the ring with nitrogen. The other rings don't have it, but these are carbon, 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 but there's a nitrogen in the ring. That's a telltale sign of an alkaloid. We all love alkaloids. I just had alkaloids for breakfast because caffeine is an alkaloid. And many important human drugs are alkaloids. Okay, so that's one thing I need you, just those little things about structure. Second of all, I want to tell you that sometimes these chemicals, which I'm going to explain are toxic, they're packaged with sugars. And I've highlighted those with green here. So how do I recognize a sugar, Dr. Karasov? Well, a ring with 5 carbons, 1, 2, 3, 4, 5, and an oxygen in it. The O for oxygen. That's a sugar. And this first compound has 1 ring of glucose, sugar attached to it. 2 on that and 3 on the other. So they could be packaged with many sugars. So keep an eye open for that kind of structure. And finally, point number 3. When they're packaged with sugars, they are rather safe, you might say. They're kind of inert. But if we strip away the sugar attachment, what remains is what's shown in the bottom row. These are that without the sugar. This without the sugar. This just without the sugar. And that makes them toxic. The first one is found in aspen trees and willow trees, and the compound is toxic to insects and parasites. However, we humans have found that that chemical is also useful to us. It's aspirin. In the second case, almonds have cyanide in them. It's usually locked up safely because of those glucoses. But if the glucoses are stripped away, the cyanide is released. Toxic. The third chemical is common in tomatoes, potatoes, eggplants, and even a weed plant we find in our gardens here in Madison called deadly nightshade. They all have this chemical, which if the sugars are stripped away, becomes toxic. So that's just a little bit about structures, natural toxins. Now, let's follow what happens to one of these when an animal eats them. We'll follow the middle one, amygdalin, that cyanide-containing compound in almonds. In the body, if the sugars are stripped away, the cyanide is released. What strips them away? In the guts of mammals, and here we're studying rodents, the small intestine makes an enzyme that can strip it away. The enzyme is used for other things by the animal. Enzymes are just proteins that do work for us. But this enzyme also happens to break the sugar off of this compound and can make it toxic. Also, there are regions in the gut of a rodent that may have microbes. I've colored them in here. In the large intestine, in the cecum, sometimes even some rodents have four stomachs. There's communities of microbes there, and they make thousands of enzymes, and sure enough, some of them will strip away the sugars from these compounds. So when a rat, we did experiments, not we, but others have done experiments. When a rat is fed amygdalin, it can be toxic to the rat. But this is interesting now. Other animals, like many of the small birds you're familiar with, American robin, cedar waxwing, have extremely simple guts. The guts don't have compartments with microbes, and their guts don't make the kind of enzyme that will activate the toxin. So when they eat amygdalin, they defecate it unaltered. It just passes through their gut. So studies, not done by me, but other people, have shown that a cedar waxwing can ingest 5 times the amount of amygdalin that would kill a rat, but has no detectable effects in the cedar waxwing. All right, you're ready now. You know just enough to be dangerous. We're going to brainstorm a hypothesis for the possible toxin in the death apples and how giant tortoises and land iguanas might tolerate eating them, whereas humans, when they eat them, get very sick. So first, let's consider what bit of information I was able to get when I came home and looked up this plant and some of its natural products. This terrible, fearful-looking compound. But I want you to look at it carefully, think of the things we just talked about, structure, and think about whether it suggests to you what might be the compound, the toxin, and what might be the name, a name of this, a group, and how maybe one animal might do all right eating it and another not. I'm going to wait a minute. You may have hunches you want to try out on your neighbor, talk amongst yourselves, and then we'll take it to the next step. Can I enlarge it? I could. I hope it doesn't wreck everything. Let's try. I'm going to escape. And I'm going to move over. Oh, I'm in a presenter mode. I have to leave the presenter mode. No, it's kind of hard to do that. Let me see if I can get back to the presenter mode. Okay. Anyone want to venture an idea, observation?
There is no nitrogen.
There's no nitrogen. It's not an alkaloid. I agree. Everyone else agree? No nitrogen. Any other ideas? Please.
I just have a question. How come, if this has been in existence in nature for eons, none of, I would venture to say almost nobody here has ever heard of it or thought of it? Repeat the question.
I couldn't quite understand you with the mask on.
If this has been a part of nature for eons, how is it that pretty much no one here has ever heard of it or thought of it? It seems not to have.
Actually, people have heard of this kind of thing and thought of it. There are analogous examples of this in our that we live with every day. Yeah.
And that tree doesn't grow here, but it does grow on St. John and the Virgin Islands where our kids live. And we've read many warnings in plant books down there. This is, don't even touch that.
Yeah. So it is no, but I will repeat what was said, okay? A member of our audience visits St. John's in the Caribbean. This tree exists in the Caribbean, and she has seen signs next to the tree that say, "Don't even think of touching me." So it is recognized to be toxic. Yeah.
So it looks like there's one sugar group
There's a sugar group.
surrounded by 3 phenolic groups that are not sugars. Do the 3 phenolic groups get stripped off? And each one of them becomes a toxin because they're not attached to the sugar anymore?
Bang, what a great idea. Notice, so the idea, I'm going to repeat the idea proposed by a member of the audience. The central unit is one of these rings with 6 carbons or 5 carbons and an oxygen. And it's a sugar, it's actually glucose. The other rings are 6 carbon rings, which we saw when we looked at willow and aspen trees, and they're called phenolics. So we've named the compound. It's a phenolic glycoside because it has a phenol ring and has sugar in it. And then, why not, maybe the reptiles can eat this, but they don't have the enzyme in their gut that strips the glucose away. It passes through undigested, whereas humans have the enzyme. When they eat this, they break it down somewhat and activate the toxin. It's a great hypothesis. I commend you all for working it out. Just needs a little more work before we publish the paper. Let's move on. There are lots of examples of this. I'll just mention that I worked with Israeli colleagues on a variety of this story, a different version of this story. We worked on a plant called the common pearl plant. And its fruit, the pulp of the fruit, contains the toxin, and the plant makes the enzyme that will activate the toxin. But it stores the enzyme in a different tissue. It stores the enzyme in the seeds. So if there's an animal that eats the fruit and chews the seeds all together, that's it. It gets what's called the mustard bomb effect from the toxin here. But there are rodents in Israel who have adapted to this fruit. They eat the fruit, they spit out the seeds. They don't chew them. So they eat the toxin, but it passes through the gut untransformed, and they spit out the seed, which has the enzyme. I'm going to play a minute of a video. Is this to clarify? Yeah, I'm going to stop there with that story. At least one species of mouse does this. Right? I'm going to play a little bit of a video that we made when we published this paper. And I'm going to stop after a minute, but it's going to be a teaser for you because I'll give you the web address and you can look it up later.
Hi, my name is Vicky. My colleagues and I found a unique chemical mechanism that promotes seed
Laos can go.
mammals in the fruit of a desert plant. Ocademus bacatus, or as we like to call it, Obi, is a keystone species mainly because of its juicy, fleshy fruits. Indeed, whenever I visit this plant, I always find a wide variety of organisms feeding on it. Seeds are an important part of the diet of a Comis carinus fluid rodent, so we were interested in studying its interaction with the Obi plant. One of the first and my personal favorite observations is of a Comis carinus approaching the Obi plant, cutting the stem right before the fruit cluster, taking the whole fruit cluster in its mouth away from the parent plant to consume at a safe place. But what happens to the fruits?
Okay, so the rest of the story, I'm going to leave it to you as extra credit. You can listen to it at home. At the end of the talk, I'll give you the web address. You can look this up. I'm sorry, we were aware we would have loved to have it louder, but we're constrained somewhat by the sound system here. So that's the best we could do. I'm going to show another video towards the end.
Would you repeat what she said? Just repeat what?
She kind of reiterated some of the points here. All right. And then introduced the animal that she studied. So, this is the end of part 1 of my talk. I just want to review our major point here. The same toxin might affect species differently, depending on their physiology and their behavior even. Some of the compounds we looked at, these compounds with glucose in them, an animal might not be impacted at all by that toxin because it has such a simple gut that it doesn't have the enzyme that will activate the toxin. Or in the case of this rodent, if you watch the video, you'll see it has a special behavior so that the enzyme and the toxin never mix together. And they're able to eat the fruit. So now I want to go on to part 2, which is an example that involves some man-made toxins. So this is about an herbicide called 2,4-D. And it's one of the most popular herbicides around the world. It's the 3rd most popular active ingredient used on plants in the United States. It's commonly used on land, spread in fields, to retard the growth, to reduce the growth of competing weeds with the crop plants. But it's also poured right into lakes to kill weedy plants in lakes that maybe make your shoreline less pleasant if you own a cottage on a lake in Wisconsin and you don't want all these weeds piling up in your shore. Your lake owners could decide, let's pour 2,4-D in the lake and kill that weed. This chemical was designed in the 1940s to act on plant physiology. It wasn't targeting animals. It targets plants. It mimics a hormone in plants, and it makes the plant grow in an uncontrolled fashion, and it goes into senescence and dies. It works really well. You might be interested in that structure. Another ring. Only this ring has chloride ions on it. Nature does not make chemicals like this, chloride ions on rings. It's a telltale sign of a man-made chemical, a man-made toxin. Now, this chemical has been around since the 1940s. Consequently, the Environmental Protection Agency long ago reviewed it to determine its risk level. And some of the studies they looked at involved exposing fish to the chemical, and they concluded that as long as the concentration in water is below 14.2 parts per million, there's no effect on fish. In fact, they coined this term, no observable effect concentration. That's what the EPA established. That's how our system works. We test chemicals and then we determine what we think are safe levels to use. The EPA would say you should not apply, and when you actually apply this in the environment, you shouldn't put it out at concentrations more than 4 parts per million. That's a safety margin. There's no effects until 14, but they want you to hold it at 4. That's a safety margin. And we also know that when people spread 2,4-D in the fields, it may come into lakes and rivers in runoff. And if you measure the levels in the runoff, it's 0.7 parts per million. So it would kind of seem that the levels that are running into our lakes and rivers are in a safe zone. And even if we follow the rules and put it in at 4 parts per million, we've protected the environment. I had a graduate student about a decade ago who was interested in this chemical, even though supposedly it was safe. His name was Zach Quattro. He designed a system where we could expose fish to the chemical. Our laboratory for this work is deep in the bowels of the Water Science and Engineering building on campus, if you know that building. It's right by the lake and right by the student union. And the system is set up to bring clean water to the fish, but then we can control how much toxin goes in. I'm going to point to some amber bottles in the figure. Right over here.
Can you describe where you're pointing because people don't answer?
In the right-hand figure, in the upper right corner, there are 3 bottles of labels. Those are the amber bottles with the 2,4-D. And the chemical is taken from those bottles and it's spread into all these tanks, although some tanks are left with what are called controls. They don't get any toxin. Now, when we started work, we designed our experiments to expose fish to very low concentrations. So remember, the EPA says the no effect concentration is up at 14. That's the top line, presumed NOAC, 2,4-D, adult and juvenile fish. And our concentrations are 0.05, that's the horizontal red line, 0.5, and 2 parts per million. All of them are actually even below the maximum allowable rate the EPA allows people to put into lakes in Wisconsin, which is 4. We grew up eggs and larvae and young fish in the water. And what we're looking for is normal development. So the 2 figures on the left are a fish, a northern pike. That's a native Wisconsin fish. Some of you have been lucky enough to catch northern pike. Those are the embryos. They're not grown and developed yet into young fish. And they're normal looking. But then we also found in our tanks some with deformities. One common deformity was what's called curved spine, scoliosis. That's in the upper right-hand figure. And the lower right-hand figure is another kind of common deformity. It's a lot of fluid around the heart, and so it's swelled up more. That's called edema. These very, very young fish will not survive. They may have been alive in the experiment, but we already count them as dead. All right, I'm going to show you a graph and how to read the graph, because one other time in this talk, we're going to look at a graph of this kind. So it's simple. These are friendly graphs. The y-axis of the graph, which is the line going up the left side, says larval survival. The higher we go up that graph, the more fish are surviving. The lower you go down the graph, the less fish are surviving. And along the horizontal x-axis is the concentrations of the toxin, ranging from 0, those are our controls, and then 0.05, our lowest concentration, 0.5 and 2. All of these are below what the EPA says is a problem concentration. These are considered totally safe. But look at what happened at 0.5 and 2 in this experiment. Survival was decreased. Not as many fish survived the experiment. This was a negative effect of the 2,4-D in the water on the health of these fish. They died. When we have a result that we can show is statistically significant, we scientists, put a star above it or an asterisk. So when you look at bars like this, and if you see a star or an asterisk, that means it's a significant difference compared to that control where there was no toxin. So my first graduate student to work on this actually made this discovery about effects at very low concentration, kind of accidentally. He was doing a different experiment, but he had some eggs left over, and he said, Bill, can I just expose the eggs to 2,4-D? And I said, go for it. And he came back later and he said, you know, this was the most dramatic thing I saw in my experiment, this little add-on. A lot of the larvae died. So we wrote a grant. And that allowed me to bring in another grad student. And he did the experiment again. So number 1 on this graph here, that's Zach's experiment, where he showed effects at all the concentrations, except the control. And here's the second study, the repeat we did with when the new grad student, Gavin, came on. These are all showing negative effects on the fish at concentrations that are claimed to be safe, according to the EPA guidelines. Now, you may be interested to know that as we began publishing these papers, the pesticide industry took note and pushed back. I remember the day Gavin called me at home and he said, Bill, 2 scientists published a paper that attacked our paper. They criticized the way we did the work. And I said, calm down, Gavin. This is good. Most of us researchers toil on doing our experiments, publishing our papers that only a few scientists in the world read. Here, we got some real attention. So I said, this is good. We're going to carry on our work. We're going to write another grant, get more money to study it. This will help. And we wrote a rebuttal that was published. How do we explain that we were finding these negative effects when they'd not been found before? We think the picture that emerged from our studies was that the very early life stages may be more sensitive to 2,4-D than young fish or older fish. And this should not be surprising because it's known for other toxicants as well. Sometimes early in development, an animal is most sensitive to toxicants because their own defense mechanisms are not fully developed. A year after we published our second paper, another laboratory published a paper on a different species of fish, and they got results just like we got. This is this number 3 study, Kehilla et al., 2019. Of significant effects at low concentrations, just like we found. Now, I'm sharing this with you to underscore the importance of replication in science. These studies are not identical studies. They all differ in certain regards, but together they advance what we might call reliable knowledge. Reliable partly because it's based on replication. All 3 of the papers I've just talked about showed significant impacts on survival at concentrations that were well below what the EPA presumed to be no effect concentration. Furthermore, with the new resources we were getting, we were able to expand our studies and look at other things. For example, we did some behavior studies with fish and made this interesting conclusion that I've written on the right side of the slide. Our results suggest 2,4-D alters the development and function of neural circuits underlying vision of larval fish, and thereby reduce visually guided behavior required for survival. In other words, the fish didn't find their food. Maybe you've heard of endocrine disruption, how toxins humans make may influence the endocrine system of animals. Now, this toxin was designed to work on the endocrine system of a hormone called the plant toxin. But when we tested the fish for their normal hormonal response to stress, looking for the hormone corticosteroid or cortisol, we found that the fish exposed to the toxin have very muted responses. This is a normal fish. This is its cortisol production and during stress. A muted response. Now, this cortisol response is a normal part of life of animals. When we get stressed, our corticosteroid level goes up, and it tells other parts of our body, get to work and address the stressful thing. The fish aren't getting the full benefit because of exposure to 2,4-D. Finally, because we are very aware from our natural studies that different species can have different sensitivities to chemicals, to toxins. So we extended our studies to include a wide variety of native Wisconsin fish species. The majority were found to be sensitive to 2,4-D, negatively impacted. So, for example, we studied 9 different species of Wisconsin fish. 7 of 9 showed these negative effects. They were in 6 different fish families. 5 of the families, 5 of the 6 fish families were showing effects. And they were in 3, we studied 4 different orders of fish, and 3 out of 4 orders were sensitive. We also realized we needed to take our experiments out of the lab and test them under more natural ecological conditions. Maybe it won't work the same way out in the wild. The water's different than our water. The other things are going on in their lives. How can we be sure this is a real ecological impact? So now I want to share with you for a few minutes some related work where we actually took this out into the field, got out of the lab, took it out into the field. And I'm going to show you a video that was just produced last year, I think, by the Water Science Institute and Sea Grant at our university. And I'll put the mic up next, and hopefully if we're quiet, you'll hear it.
We are looking at the effects of 2,4-D, which is a common herbicide that's used in our lakes around here to control Eurasian water milfoil. Eurasian water milfoil is a very nasty invasive species that creates these mats all over the surface. And we're looking to see how this herbicide impacts the early life stages of native Wisconsin
We
fish.
are looking at the effects of 2,4-D, which is a common herbicide that's used in our lakes around here to control Eurasian water milfoil. Eurasian water milfoil is a very nasty invasive species that creates these mats all over the surface. And we're looking to see how this herbicide impacts the early life stages of native Wisconsin fish. And we're focusing today pretty heavily on fathead minnows. This is a very unique herbicide. Most people think herbicide, they think adding it to the agriculture world, adding it to crops, but this is a herbicide where we're actually putting it into our lakes. We do a lot of this work in the laboratory, so we kind of designed 2 sets of experiments that utilize more natural lake water. We have what's called a mobile exposure system that takes water directly from the lake, brings it up into a head tank, mixes it around, and then distributes it to the rest of the tanks where all the fish are held. And then we have an in situ or an in-lake exposure system. And this is an exposure system that's built basically with 2-liter buckets with holes in them. We mimic it then to what goes on during an actual active herbicide treatment. The DNR permits 2,4-D into this lake. They will go and apply 2,4-D to this whole entire lake. It'll kind of equalize to a certain concentration, and then we will see kind of how that herbicide treatment affects the 2 systems that we have going on. The really cool thing about this project is we get to actually work with lake associations that are doing the 2,4-D treatments themselves. So overall, what your goal is to build this risk assessment guide that says, okay, if you want to treat this invasive species, you can treat with X herbicide, or you could do Y or Z, and all of it kind of gives you this little idea of which is the best way that we don't hurt our environment.
We're syncing. The Zoom people are just finishing the hearing the video.
I just unmuted.
We good? Okay, so now I'm putting you back to work. Gavin, that second grad student, he's actually gone on to be a scientist at the Water Science Institute and Sea Grant. He's done at least 2 experiments now where he looked at lakes in Wisconsin where the homeowners association decided to add 2,4-D to the lake. So will the survival of fathead minnows be reduced under an actual routine herbicide treatment, the way the EPA says it could be done safely? I'll pause for a minute, share your idea, your answer to that with your neighbor. Curious what people are thinking. Oh my God, how are they ahead? Okay, I gotta give the answer because they know it already. Yes. Okay, again, this is that graph where the taller the graph, the better the survival, the shorter the graph, the poorer the survival. And in the lakes that were treated, in the gray, lower survival when the same lake, the experiment was run without treatment. That's a kind of internal control. Gavin mentioned in his video these lakeside associations, lake homeowner associations. Gavin's done a lot of traveling around the state talking to homeowner associations to get them to reconsider their decisions about using 2,4-D in lakes. And there have been a couple times where he's not been able to go and I've been recruited to do it too. They're very interesting to go out and talk with people living on the lakes who want to get rid of these weeds, and they have to struggle with the choice of should we use this herbicide. All right, so I want to finish up just reviewing the couple major points we focused on today. First, with that first part of the talk, we learned that the same toxin might affect species differently depending on their physiology and their behavior. And we even used that principle when we designed our experiments with 2,4-D, because we tested lots of species of fish. And what we learned is a commonly used herbicide, 2,4-D, that was designed to work against plants, not animals, still has surprising negative effects on fish during early development. So I'm going to stop my talking. You now have a sense of what maybe a physiological ecologist does, mixing physiology and ecology. And I'd really be happy to hear questions you have. And we'll start on the left and move to the right, please.
So if application of 2,4-D reduces the survival of fathead minnows, there are fewer of them in the environment. There are fewer fathead minnows eating whatever fathead minnows eat. Are there then ripple follow-on effects to the lake and the ecosystem because it changes the balance of nutrients and animals?
So the question is, what are the downstream ripple effects of having 20, 30% of small fish like fathead minnows die, or all the fish in early development dying, or most species? And other kinds of ecological studies that look at interactions in lakes, people would say, well, this could lead to a reduction in the number of, ultimately, the top predators, which are the fish that human beings, the lake owners, they're fishing for them, they want to get them. There'll be fewer fish in the lakes, that's one possible effect. And that's just something that makes sense to us. But another thing we learned from ecology is there are often other ripple effects that we can't so easily predict. It's a very complex system there in the lake. And there may be other kinds of effects. And I'm not doing work on that, so I'm hesitant to speculate. But your speculations were nice, and some of you might have some other ideas. Let's move on to the next question. Back.
One of the things I'm wondering about in there is, what is the effect of this getting into other water systems, going down through our basic water system, and does it affect the larger lakes? Are we somehow poisoning lake after lake in the water?
So when we pour this toxin in the lake, the concentration is at its highest right after application, and then it goes down over weeks to months, down like that. So it's not there forever. It will go down. Very, very large lakes are too big to treat, and maybe isolated bays sometimes are treated on very large lakes. But other times the lake is within a size that these boats can travel back and forth through the lake with the chemical pumping out, and the whole lake is brought up to a concentration of several parts per million, which will then, again, over months decline. But during that time it's declining, our studies show the levels are high enough to kill larval fish and affect their ability to find their prey and affect their hormone system. I couldn't even guess all the possible repercussions in their physiology. We've only studied a few things.
You said that it gets less and less, so what is it? It's breaking down?
Yes, it breaks down. Chemicals in the environment, even the ones we make, break down due to exposure to sun, due to microbes sometimes in the water system, due to other features of lake chemistry. So for a lot of reasons, they break down over time. The chlorinated hydrocarbons, which I introduced to you as compounds where there's a chloride or multiple chlorides on a ring, they're very resistant. So they hang out a long time. And some of those chemicals hang out so long that they accumulate in the body of animals in the waterway. And that's how you end up with fish or marine animals with very high concentrations. Other animals eat them, and the chemical can be concentrated as you go up a food chain. That does not happen with 2,4-D. We've not studied that, but other chlorinated hydrocarbons.
So you've shown us some of the unfortunate things that happen to fish as they develop in their infant stages and on, or maybe fetal stages, whatever, as a result of exposure to this herbicide. I'm wondering what kind of studies or results we know about with humans who may live near fields that are treated by this herbicide or live along waterways that are also treated with this herbicide. What do we ourselves have to be cautious about?
Great question, and I'm going to repeat it. What are some of the effects on humans if they come into contact with this chemical when they're spreading it or when they're swimming in the lake? Who knows when?
And especially in utero or...
Oh, okay. So I will say this for our EPA guided testing system. We do the best job on impacts on humans. You know, who are we most interested in the world? Ourselves. So we do a better job of testing for that. And there have been many layers of testing. And we know that 2,4-D, if it's handled properly with protective gear on, and the dosages are controlled, that we can keep it relatively safe for the people that apply it. Although I would have to say, I wouldn't want my career to be applying any toxic chemical and getting a lot of potential exposure. But it is, we know levels that are relatively safe. And when we've looked for impacts on humans, you don't see a lot of reports of impacts of 2,4-D in the lakes on humans. Because I was going out to speak to some of the lake owner groups, I did a little reading, more extensive than I would normally do, looking for biomedical implications of the compound. And I really didn't see a lot. It can cause an itch in some people, but that's not a major problem. But in doing that reading, I learned some other things, other huge gaps in knowledge. And again, they're not about sensitivity to humans, but as much as it's, I've made a case that this is bad for fish, there have been very few studies of the effects of these chemicals on the invertebrates in our waterways. Insects, shellfish, and so on. I'm going to keep.
You said it was, you repeated the word relatively, the words relatively safe. So I'm not quite sure what you meant by that.
What do I mean by relatively? That's one of our squishy words, because so often we don't know things in absolutes in science. So, relative to some other things that we know are really bad, 2,4-D doesn't look so bad. Okay, moving along, yes.
It's really interesting to know as much about the downside risks of things as possible. What about the downside risks of not using this? Do we know anything about what happens to an ecology when these weeds completely overgrow or get out of control? So we can what is the good stuff?
That's a great point. Why are we using these chemicals in lakes in the first place? Now, often the concern by the people who make the decision to put it in a lake is because it's a nuisance. It's a nuisance to their motorboats because it's a very stringy vegetation. It's Eurasian milfoil. That's the name of the plant. Or it's polluting their beaches. So that's one kind of risk of not treating it. Will it overrun a lake? And let's imagine worst case scenarios. It draws a lot of oxygen out of the water. And so fish are dying anyways. I don't think that's been reported. It can shade out a lot of plants that are submerged because it blocks sunlight. So I don't think there's any argument that the aquatic milfoil, as you suggested in your question, will have its own kinds of effects on the ecology of the lake. And I am not even going to argue that we shouldn't try to do something about it. We might want to do something about it in some cases. And that's where we have to use our smarts, our knowledge, and our intelligence to pick ways of using it that cause the least other secondary effects. And that's why we go talk to the lake owners, because we can give them suggestions. Number one, maybe you want to use this chemical, but how about let's not use it when the fish are laying eggs in the late spring and early summer. Let's just wait a little later in the summer, because as the fish grow up, they become tolerant to the chemical, and it doesn't affect them so much. Or are there competing products that could also inhibit the growth of the milfoil plant, but maybe don't have the same effects? We've actually been simulated to do some work on some other products that we have found to be a little less potent than 2,4-D.
Bringing it local, the city of Madison, I know, cuts weeds on the lakes. Do they do spraying or?
I don't know. That's another alternative. Sometimes the lake associations hire boats that will skim across the lake and collect this weedy material and dump it on land somewhere. So they're removing the plant without applying chemicals in the environment. And yes, we see I've seen boats of this sort in Monona Bay doing that kind of thing. I don't know if the city is using any 2,4-D. I don't know. Does anyone else know? Another question.
Two questions. Is this one of the chlorinated hydrocarbons addressed by Rachel Carson in Silent Spring? And then the other one is, have you looked at whether if you keep subjecting a population in the lab, for example, to 2,4-D, do they become resistant to it? Can you unnaturally select for fish whose larvae are not sensitive?
Okay, two questions. is this kind of chemical, like what Rachel Carson and other people have written about so-called persistent organic compounds that tend to stay around in the system and even concentrate in animal tissue. That was the first question. And the second question, which one we should get to, remind me again, was?
If you subject.
Okay, can species adapt to it? That was, so let's go with the first one. You might still have to remind me of the second. So, it is, by chemical definition, a chlorinated hydrocarbon. However, does not have some of the hugely negative effects of the kinds of chlorinated hydrocarbons that Rachel Carson wrote about. She was writing about DDT. Others, after we figured out DDT and decided to stop using it, another group of chemicals called PCBs, polychlorinated biphenyls, they were used until we thought, oh my gosh, they're doing the same thing DDT did. So we stopped using that. And we have other problems with chlorinated hydrocarbons, dioxins, you may have heard. So our society has gone through a series of these chemicals, and we will continue. And all we can do is keep our minds open, study, looking for effects, because the names may change, the chemicals may change, but the problems remain. Now, can species adapt to toxins? And there are some examples where in heavily polluted environments, especially environments polluted with metals, heavy metals, where organisms have adapted and can come to tolerate more of the heavy metal. We know the mechanism by which they do it. They actually make more of an enzyme in their body, a protein really, that binds the metal. So what they're doing is they're exposed to a lot of metal and they bind it to this protein in their body and it accumulates in their body, but it can't have the toxic effect because now it's bound up with a protein. So there are a few examples, and that's one where animals have adapted. The most classic example of adaptation by natural selection to toxic chemicals is what insects are doing all the time. We develop new sprays and toxins, we put them out on the fields, and within 1 or 2 years, the few species that have a mutation that allows them to survive, they reproduce a lot, and we have a new genetic population of the insect, which is tolerant of the toxin. So it can happen. And it happens fast in insects because they have very short life cycles, because it depends on multiple times of reproduction, production of new generations. We don't see this kind of adaptation so much in vertebrates because they stretch their lives out so much, it can't happen fast.
So you don't see the 2,4-D in fish?
So I have one comment just to what Francie was saying after 12 years of chairing human subject committee. To study effects of toxins on human is probably impossible. Human subject committee would allow controlled studies. We remove babies and expose them to 2,4-D, and another group no.
So
the second problem is that humans need to know. That's why fruit flies, for instance, are so popular for research. Their life expectancy is so short, and I assume with species the same thing. But I also have one question I wanted.
Let me just reframe for people who may not have heard it in the Zoom audience. We have a member of our audience who's pointed out that it's actually hard sometimes to do the testing in humans because we don't want to expose in the humans deliberately the chemicals and look for effects. And actually we have model organisms and even tissue cultures sometimes, which help us, we can expose those to the chemicals and it helps us predict what would likely happen in the human. And we do have epidemiology studies where we see populations that have been exposed and compare them to populations that haven't. And that informs, I think. Okay, so now a question.
Yeah, the question was when you mentioned that EPA established the toxic level and the safe level, what kind of studies are they doing?
What did they do? Okay. This was very disconcerting to me when I looked into this. They did a few studies of their own, and actually the design of our water system, we took from the EPA laboratory in Duluth. My grad student went up there, studied with them for several weeks, came down and then built a similar system in Madison. So they do some studies, but they were doing them with often young and adult fish. They weren't doing them with the early stages. But when we got that critical article, the critical article, and it was written by two people in the pesticide industry, it mentioned, well, there have been studies on young fish, and they didn't see what you're seeing. And so I looked up to see, we've not heard of any such studies. So I looked at what they cited. They cited unpublished studies by the industry. There's no way I could ever look at the data from those studies. So they're making the argument, we've done that and we didn't see it, but you can't see it either. So EPA relies sometimes on these on studies that have not been peer reviewed by independent scientists. It's an unfortunate part of our system.
So I grew up in a farm in southern Minnesota, and I recall my father using 2,4-D on his crops. But even before I left, many years ago, the pivot from that was to Roundup. And I was curious if you had any insight or comment on whether Roundup, which was advertised at that time, and I think even today, that it breaks down very quickly in the environment and is therefore non-persistent and better herbicide in essence.
So I think Roundup has glyphosate in it, doesn't it? Does anyone know? It's glyphosate, okay? So Roundup was a next generation herbicide.
Right.
Safer, okay? Well, the studies that have been done looking at when glyphosate gets in aquatic systems, it has big negative effects. I know there's been a lot of studies on amphibians. I'm not sure about the fish. Atrazine, another herbicide.
Also, my dad used that too.
Okay, yeah, these are designed against plants. But we learned that atrazine was a gender-bending chemical in many animals. Again, some of the research came out in amphibians. So that very low levels that everyone thought was safe, were causing amphibians to come out all one sex. So we try to do better. We even do our best to test these things, but I guess you can take away your own conclusion from this kind of history. Other questions or comments, please?
This goes back to part one, when we were talking about the little death apple tree. And I was wondering if studies have been done that show interspecies differentiation so that humans may respond differently, maybe not to that particular toxin, but to the toxins in general.
Yeah. So could there be difference within a species, but maybe populations living in different places? Could there be different responses? And maybe, we could even think of humans perhaps. So I'm going to now totally speculate. All right, so allow me to speculate. And even looking within humans, okay? So first of all, one way humans circumvent these chemicals, which believe me, these toxins are in all the food, so many, not all, so many. But we process these foods, we cook or soak them in different ways. And in different cultures, in different places in the world, they've come up with different ways of dealing with toxic chemicals. Okay? So that's a kind of inter-population difference. But now, and here's my speculation. One of the kinds of enzymes that mammals make that can break the glucose away from these toxins, it's in a class of enzymes called beta-glucosidases. We make one. It's called lactase. It breaks down milk sugar. It's part of our biology. But research has shown that this very same lactase can do this to some of these kinds of toxins and break them too and activate the toxin. And we know among humans that there are populations with very high lactase levels in the population and other populations very, very low levels. And I'd predict that there might be some differences between those two kinds of populations and how they respond to some of these kinds of toxins in their food. Total speculation. Okay, another question.
So does this mean, with all this interference of pesticides and such, does this mean that, are you sending us a message that we should all buy organic now?
I am not sending you that message,
Okay.
right? I am not. Let's start on the left again and sweep.
Do you feel that the basic confidence in science as a way of finding answers to difficult and sometimes politically controversial questions has been under attack? Maybe that's a softball question. What do you think about that after COVID and COVID?
Well, yes, we do see instances of people foregoing consideration of science. Science is a way of thinking. It's a process we use to test ideas and see which ones can go forward with support and which ones are unsupportable. When you throw out the scientific method, you don't have a strong leg to stand on for conclusions. The scientific process is our friend, and when we don't pay attention to it, we are living in more ignorance. So, is that the kind of thing you wanted me to say?
I'm interested in how you feel about it.
All right, but I'm going to give you another viewpoint. I'm impressed that our society had recognized early, we recognized, let's say, we recognized early enough the problems with DDT. And we controlled ourselves. We made laws, we stopped the production and use of it for the most part, before we destroyed it. Then something else came up, PCBs, I mentioned. And we went through the same process, but we stopped it once we realized. Many of you remember acid rain, a very serious problem, but we learned about it through science, and we came up with a way to deal with it, and we did it as a society. We are smart people. We have great ability to figure things out and act on it, but it's only when we embrace this scientific method and pay attention to it and not just make claims coming off the top of our heads.
Talking a little more about what I was saying, when we know that the EPA went through a huge renovation when it reduced that down on the previous administration, we realized that many of the new EPA people do not necessarily have the same level of training. And there are a number of organizations which are very critical, saying that when it comes to doing studies on human health, that a lot of it is going back in time, that a lot of parts of the research, such as looking at endocrine disruptors. Endocrine disruptors are, that a lot of endocrine disruptors research is not actually followed up. And it's not included. EWG, there are a number of organizations that complain about it. It took us years when we started out looking for drugs for cancer. If we only looked for cancer effects, we didn't look for the effects of drugs on other parts of the body. When we look up effects of 2,4-D on humans, there are many, many studies that are not included in EPA's work, such as right now looking at effects of 2,4-D on farmers. The difficulty of including it is when there's a high rate of lymphoma and other cancers in farmers who work with 2,4-D. The question is, how do we get past this? Because we have a lot there. Who makes something in ecology? And there are so many variations, and so many other possibilities that you can't get an absolute proof, like cigarette smoking. So how do you do it?
How do we deal with uncertainty? We just strive and do the best we can, accepting that we don't know everything and being open to ideas, and when we can afford to do it, testing things. But yeah, we will never 100% solve these problems. Let me see if there's any more questions. And Barry, do we need to tie things up?
Tie things up,
Real
but
quick
we
comment.
don't want to limit you as long as there are still questions.
The comment is that in the long run, what makes it work is courage. People like you, who publish, even though you know it, anger, people who have substantial interests. Thank you.
Oh, thanks. Thank you.
Thank you so much. I got very nervous when you mentioned Dutch Hollow Lake. Sure.
I don't want to look at anything, but when Bill talks about the studies in Israel and race about the mice, some that spit out the seed. You look at the study, you'll see it's just there's another box that does the opposite. It spits out the fruit, the mushy part of the outside, and eats the seed.
Nature is always amazing to me. My local beavers have destroyed, I don't know, about an acre of willow, a pair of them over the winter. How do they keep eating that, all those phenols?
I'll talk to you about it on the side.
Thank you, everybody, for your attention and questions.
Thank you so much. It was so informative and a little bit scary. Did you look at, does it affect the peepers in the lakes as well, or do they add it later in the spring?
I've not worked with them. I can't say.
Yeah, if you want to see that Israeli study that we participated in, just type this in Google, and you'll come up with that video. It runs about 4 minutes, I think. Oh yeah, you can email me too.
It's my friend is about 8 miles south of Dutch Hollow Lake.
Do you know people who own property?
Oh, yeah.
Oh, thank you very much.